Amino acids are structural units that make up proteins. As its name suggests, amino acid consists of an amino group and a carboxylic group, along with a hydrogen atom, all of which are attached to a central carbon known as the alpha carbon. Amino acids differ from each other in their side chains, or R groups, which vary in structure, size, and electric charge. Under physiological conditions, the carboxyl group is deprotonated and carries a negative charge, whereas the amino group is protonated and carries a positive charge. Each amino acid has both a 3-letter abbreviation and a unique 1-letter symbol. The 20 common amino acids can be categorized into 5 groups based on their side chains. Both nonpolar amino acids and aromatic amino acids are hydrophobic. There are 7 nonpolar amino acids abbreviated as GAVLIM. G stands for glycine, also abbreviated as GLY, which is the smallest amino acid with a single hydrogen atom as the R group. A stands for alanine, abbreviated as ALA, which has a methyl group as its side chain. V stands for valine, abbreviated as VAL, which has a 3-carbon isopropyl side chain. L stands for leucine, abbreviated as LEU, and I stands for isoleucine, abbreviated as ILE. Leucine and isoleucine are structural isomers of each other, both with a 4-carbon side chain. Together, valine, leucine, and isoleucine are known as the branching amino acid, abbreviated as BCAA. M stands for methionine, abbreviated as MET, which is one of the 2 sulfur-containing amino acids, with 3-carbon thioether side chain. P stands for proline, abbreviated as PRO, which has a distinctive cyclic side structure with a secondary amino group known as imino group. There are 3 aromatic amino acids abbreviated as FYW. F stands for phenylalanine, abbreviated as PHE. As its name indicates, phenylalanine consists of alanine attached to a phenyl group, also known as an aromatic ring. Y stands for tyrosine, abbreviated as TYR, which has a phenyl side group, indicating the presence of alcohol attached to the phenyl group. The phenyl group has a pKa value of 10, which means that under physiological pH of 7.4, the phenyl will remain protonated. Recall that pKa is the pH at which the compound is half protonated and half deprotonated. The higher the pKa, the higher pH is required to deprotonate a proton. W stands for tryptophan, abbreviated as TRP, which has a unique side chain consists of a pyro ring fused to an aromatic ring. The fused double ring is known as an indole. Polar, acidic, and basic amino acids are hydrophilic. There are 5 polar uncharged amino acids abbreviated as ST, C, and Q. S stands for serine, abbreviated as SER, and T stands for threonine, abbreviated as THR. Both serine and threonine have a hydroxyl side chain attached to 1 and 2 carbons respectively. C stands for cysteine, abbreviated as CYS, which is the other sulfur-containing amino acids besides methionine. Cysteine side chain contains a carbon attached to a thiol group with a pKa value of 8. Therefore, the thiol group remains protonated under physiological conditions. Cysteine is a weak acid that is readily oxidized to form disulfide bonds, which play a special role in structure of many proteins. N stands for asparagine, abbreviated as ASN, and Q stands for glutamine, abbreviated as GLN. Both asparagine and glutamine have amide side chains attached to 1 and 2 carbons respectively. There are 3 basic amino acids abbreviated as KRH. Basic amino acids are protonated under physiological conditions, therefore they are positively charged. K stands for lysine, abbreviated as LYS, which has a 4-carbon side chain attached to an amino group with a pKa of 10.5. R stands for arginine, abbreviated as ARG, which has a unique side chain known as guanidinium with pKa of 12.5. H stands for histidine, abbreviated as HIS, which has an aromatic imidazyl group as side chain with a pKa of 6. Therefore, histidine is uncharged at physiological conditions. Due to its pKa being close to 7.4, histidine acts as an effective catalytic residue, facilitating many enzyme-catalyzed reactions by serving as both a proton donor and acceptor. There are 2 acidic amino acids abbreviated as DE. Acidic amino acids are negatively charged because their side chains are deprotonated under physiological conditions. D stands for aspartate, abbreviated as ASP, and E stands for glutamate, abbreviated as GLU, both of which contain carboxyl side chains with pKa values of 3.65 and 4.25 respectively. Note that asparagine and glutamine are simply the amide versions of aspartate and glutamate. Besides the 20 common amino acids, there are several non-standard amino acids. 2 non-standard amino acids are derived from stop codons. The stop codon UGA can code for the 21st amino acid selenocysteine, abbreviated as SEC, and the symbol U. The stop codon UAG can code for the 22nd amino acid pyrrolysine, abbreviated as PYL, symbolized by the letter O. In addition, special amino acids ornithine, citrulline, and argininosuccinate are formed as intermediates of the urea cycle, which degrades nitrogen-containing compounds. All 20 of the common amino acids are alpha amino acids, meaning that they have a carboxyl group and amino group bonded to the same carbon, which is the alpha carbon. The only naturally occurring beta amino acid is beta-alanine, in which the amino group is connected to the beta carbon with respect to the carboxyl group. Beta-alanine serves as a component of coenzyme A derived from vitamin B5. For most amino acids, the carbon atom is attached to 4 different groups, making it a chiral center, allowing the conformation of 2 optical isomers, the D conformation and the L conformation, which are non-superimposable mirror images of each other. The amino acid residues in protein molecules are exclusively L stereoisomers. Glycine is a special case since its R group is the hydrogen atom. Its alpha carbon is achiral. On the other hand, isoleucine and threonine possess a second chiral center in their R groups, allowing each of them to have 4 optical isomers. A common way to determine protein concentration is through spectral photometry, which measures the amount of light that can pass through a solution, which is due to the ability of aromatic amino acids to absorb ultraviolet light. At the wavelength of 280 nanometers, the absorbance of tryptophan is a lot greater than tyrosine, whereas phenylalanine doesn't really contribute to the absorption spectra. Amino acids are amphoteric, meaning that they are capable of acting as both an acid and a base due to their amino and carboxyl groups. Each amino acid has a characteristic titration curve. Recall, in chemistry, acid-base titration involves the gradual addition or removal of protons. Take the titration curve of glycine against hydroxide anion as an example. The horizontal axis refers to the equivalence of hydroxide anion added, and the vertical axis refers to pH. Glycine has 2 pKa values, one at 2.34, which corresponds to a carboxyl proton, and the other at 9.6, which corresponds to amino proton. At very low pH, the predominant ionic species of glycine is the fully protonated form, which corresponds to a net positive 1 charge. As half equivalence of hydroxide ion has been added, the pH reaches the pKa of the carboxyl group. At this point, glycines with protonated and deprotonated carboxyl group are in equilibrium, which corresponds to a net positive 0.5 charge. When 1 equivalence of hydroxide ion has been added, the pH reaches 5.97, which is the midpoint between the 2 pKa's. At this point, the carboxyl group is fully deprotonated, while the amino group remains fully protonated, resulting in a net charge of 0. This characteristic pH is known as the glycine's isoelectric point, abbreviated as Pi. As additional half equivalents of hydroxide ions are added, the pH rises to 9.6, which corresponds to a pKa of the amino group. At this point, glycine with protonated and deprotonated amino group are at equilibrium, resulting in a net negative 0.5 charge. Lastly, when 2 equivalents of hydroxide ions have been added, the titration is complete, with both the amino group and the carboxyl group fully deprotonated, giving glycine a net negative 1 charge. The buffer zones of glycine occur within plus and minus 1 pH of the 2 pKa's, which can resist changes when either acid or base is added.